So the first question, as I mentioned at the beginning of the lecture, of the last video, the first example is the using pesticide in farmland that will produce insect or bugs that is resistant to pesticide. Second example is the overprescription of antibiotics that will produce the antibiotic resistance bacteria. And then the second question, do we have more cells or more microorganisms living in our body? The answer is we have more microorganisms living in our bodies than the number of cells we have. So if we disrupt the microbial community, that means the microorganism living in our body, what will happen? It can increase our susceptibility to infectious disease. That means it is easier for us to get sick, or it is easier for us to develop cancers, or it can be we can develop allergy. We can have irritable bowel syndrome, Crohn's disease, autism. So the question here is that the microorganism living in our body with us, do they have a mutualism, compensalism, or parasitism with us? Well, I'll tell you this answer. I'll tell you the answer to this question at the end of this video. You can think about this. So a major episode in the history of life. The Earth is formed about 4.6 billion years ago. Some textbooks said that it is like 6 billion years ago. And then prokaryote. The cell does not have a true nucleus. That means the nucleus there's no nuclear membrane surrounding all the chromosomes or all the DNA. Occur around 3.5 billion years ago. And then oxygen production around 2.7 billion years ago. Why? Because there is the autotroph or prokaryote that can do photosynthesis. As soon as prokaryote can do photosynthesis, then oxygen starts to accumulate on the earth. And then this situation, that means prokaryote and autotrophic prokaryote, they live by themselves by 1.7 billion years, and they continue until now. Eukaryote, well, believe it or not, we have a true nucleus, meaning that our DNA is surrounded by a nuclear membrane. We also have other membrane-bound organelles, such as lysosome, Golgi apparatus. Eukaryote evolved from prokaryote. And here we talk about the mitochondria and chloroplast. We believe that mitochondria, they are descendant of the smaller prokaryote, and chloroplast, they are descendant of plants or algae, or also prokaryote. It's just that they somehow get incorporated or live inside the eukaryotic cell and develop something called mutualism. A mutualism means that it benefits each other. A relationship that benefits each other. We call it symbiosis. So, what kind of relationship is that? Mitochondria. When mitochondria live inside a eukaryotic cell, the eukaryotic cell can provide protection to the mitochondria, and also the eukaryotic cell can also provide a nutrient to the mitochondria. And mitochondria, in return, mitochondria can provide energy, ATP, to the eukaryotic cell. That's why we call it a mutualism. It's a win-win situation. They benefit each other. So how does life begin? How does life begin? How does life begin? Okay, it's not, everything starts with the synthesis of simple organic molecules such as amino acid, nucleotide, monomers. The simple organic molecule we're talking about are the monomers that we talk about at the beginning of the semester. Remember we talked about the 4 major biological molecules, or 4 major macromolecules. Carbohydrate, the monomer of carbohydrate, they are glucose, fructose, and galactose. And then the monomer of protein will be amino acid. The monomers of lipid will be fatty acid and glycerol. And the monomers of nucleic acid will be nucleotide. So the first step is to make all these monomers. And then after that, the second stage is to connect all the monomers to become a polymer. But at the beginning of the semester, we told you that we use enzyme to do this process, to speed up this process. But at the beginning, we use abiotic synthesis, meaning that joining all these monomer to become a polymer without using enzyme. So we will tell you how can we join this polymer, how do we join this monomer into a polymer without using enzyme a few slides later. Let's go through the 4 stages. The third stage is to form the pre-cell. As you know that if we are able to form the polymer, it will not be easy. So what we want to do is that we want to keep the polymer in a stable environment, so that the polymer will not break down so easily. Then we form something called a pre-cell. We use a membrane. We use membrane to wrap around these polymers so that it will be isolated from the external environment, because external environment, they are not stable, it changes all the time. So by doing that, you will keep the polymer relatively stable. And then we have this, the last one is the self-replicating molecules. The keyword is inheritance. Basically, after we form the pre-cell, we want to continue the pre-cell. How do we continue the pre-cell? By developing some kind of a molecule or mechanism that can replicate the pre-cell and also replicate all these more polymer inside the cell. So we go through each stage one by one in details. So at the beginning of the Earth, we only have water, methane gas, ammonia. So the question is, what are the 4 most abundant elements in human body? Do you still remember? At the beginning of semester, I told you that CHON, right? Remember CHON? C-H-O-N, CHON. It tells you the 4 most abundant element in our human body. Coincidentally, CHON, C-H-O-N, carbon, hydrogen, oxygen, and nitrogen, they are the elements that already exist at the beginning of the early atmosphere of the Earth. H2O, methane gas CH4, and ammonia NH3. In other words, the 4 most abundant elements, they are the primitive element exists in the early atmosphere of the Earth, coincidentally. Yes. And based on these 4 most abundant elements in our body and also in the early atmosphere of the Earth, then we can form the monomers of the 4 basic biological molecules. So this is the experiment that people do to replicate, to try to simulate and replicate the early Earth atmosphere. So on this end here we have water, and then it is being heated up, and then the water vapor will travel to the atmosphere. And here we have the gas, methane gas, ammonia gas, and hydrogen gas. And then water vapor will mix with all these gases. And then we have the electrode here to simulate the lightning here. Oh, here, it simulates the sea. And this is the volcano under the sea. They will heat up the seawater. And then here mixed with all the water vapor, H2O, will mix with all the gas. And then we will simulate the lightning, and then we have the condenser here. The condenser here simulates the cold temperature. The cold temperature will condense the liquid that is being formed in here. And then we collect the liquid here for chemical analysis. So what happens is that if we keep doing this for a week, then we actually form some kind of monomers from the collection of the fluid here. We get some monomers here. The guy who do this is Miller, and many laboratories has repeated Miller's experiment, and they are able to produce organic compounds. Remember our first lecture, scientific method. If one person successfully finish an experiment, then other lab will repeat, see if they can also do the same thing, and they can get the similar result. Now, after we get the monomer, how do we connect the monomer to become a polymer without using enzyme? So what happened is that they thought about this. They use this monomer. They drip the monomer onto hot sand, hot clay, or hot rock. The heat on the rock, on the sand, on the heated surface, they will speed up the reaction. They will provide enough energy to connect the monomers into polymer. Okay, question for you to think about. After polymer or macromolecules are formed, what is the next step? Yes, it is open book, open notes. You can refer to your textbook. And what would be a reasonable explanation for the next step? And in the next video, I will tell you the answer. And remember, at the beginning, there's a question. The question is asking you, the microorganism living in our body, what kind of relationship we have with those microorganisms? The answer is mutualism. It is a win-win situation. The microorganism living in our body, we give them the nutrient and also the kind of relatively safe environment for the microorganism to live. And the microorganism help us to keep us away from other diseases, cancers, or allergy. So that is the mutualism. So think about these two questions, and then I'll answer these two questions in the next video.